In this video, we will discuss about what are the advancement in the CAR T cell therapy occurs so far. So, how these CAR T cells, they or we can say chimeric antigen receptors, uh, are being introduced into the cells, or in other words, how it is selected that which cell is suitable for this chimeric antigen receptor or which T cells is suitable. It depends on the T cell population and whether, and also depends on its commercialization. If there is if someone selects rare T cells, then it will be difficult to scale up and commercialize. That's why one of the most popular T cells that is used for chimeric antigen receptor is this CD26 high T cells. Why? What are the advantages of this CD26 high T cells? Is that it replicates the function similar to the stem cells. It releases high cytokine production. That is, immune response is high whenever it detects tumor cells that has a similar antigen as chimeric antigen receptor, and it has stem cell like properties that I have already told, like it will activate beta actin, LEF1, and the trafficking of these CAR T cells uh, is high compared to the uh, CAR T cells that are produced in other T cells. And this is the representation of how this CD26 high T cells will respond uh, whenever there is a chimeric antigen receptor activates within those CD26 high T cells. These are the responses that it will release, that is IL-17A, IFN gamma, IL-2, IL-22, and that will helps in uh, that helps in killing those tumor cells that have been directed by these CD26 high CAR T cells. Another cell system that is used is gamma delta T cells. The advantages of this is that it helps in co-stimulation and antigen presentation. It is this kind of cell system, CAR T cells, helps in producing this function, that is normal function of T cells, that is it detects the antigen of the tumor and helps in presenting those antigen to the other immune cells to act so that they can uh, release the cytokines that helps in killing those tumor cells. However, the problem with this kind of cells is that the exhaustion is reduced, such as this PD1 and TMI3 uh, signaling get reduced in the case of this gamma delta T cells compared to the other CAR T cells. These are two uh, common cells, T cells, that are being used for this chimeric antigen receptor. And now the panel on the right hand side represents some of the advances, or we can say synthetic universal CAR T cells. The first one is this supra CAR cells. So it is similar to the CAR T cells that we have discussed in our previous videos. That is, it has this antigen that is generated by immunizing animal a humanized animal against the tumor antigen, and this is together it is the mixture of it is a combination of variable heavy chain and variable light chain, and it is called single chain variable fragment. So what happens in this supra is that this antigen can be switched so that it can target different so that it can this single chain fragment can be switched so that it can target different antigen, as it can be seen here, and this switching can be occur with the help of this zipper. And another version of this supra is that it can target the multiple antigen uh, with the help of this zipper, that this combinations of this zipper changes so that interaction of this zipper changes so that it can target multiple organ with multiple single chain fragments. And another modification that happens in this kind of cells is that this intercellular domain is also modified to release the signals uh, based on the strength of the extracellular extracellular bonding or interaction. So In this way, they can stimulate or control. They can stimulate or control the kind of immune response that should be released whenever this single chain fragment interact with any tumor, interact or you can say detect any tumor that has the antigen that can be detected by this single chain fragment. And this tunable part is shown in this side. And this part is about this figure is about the switching target with new single chain fragment with the help of zipper, and the panel on the right hand side is about tuning signals through this zipper binding affinity. And in this case, there are multiple single chain fragment is there with multiple zipper. So what happens uh, based on the number of antigens, based on the amount of antigens it detects, the signaling gets the downstream signaling or intercellular signaling gets activated according to the strength of this antigen and the tumor interaction. If there is only one antigen and tumor interaction, signaling will down intercellular signaling will activate to a smaller extent. If there is the antigen get in both the antigen detects the tumor antigen, then downstream signaling will activate to a stronger extent. It will release a higher immune response or cytokines. The last one is about the anti-tag CAR T cells. In this case, what happens is that instead of directly targeting the antigen of tumor cells, the antibody has been released. Antibody has been antibody has been used to attach the antigen of the tumor cells and with some fluorescence signal, and the CAR T cells then can be used to target those fluorescence signals, so that whenever the fluorescence signals get detected, it will release the CAR T cells will release the immune response against those tumor cells. So this approach is shown here, that this antibodies will be able to target these fluorescence of the antibody that are being used to target this antigen that are present on the tumor cells. So in that case, what happens is that whenever this antibody of the CAR T cells detects this fluorescence, it will release an immune response that can kill those tumor cells. So these are some of the advancement in the CAR T cell therapy to take care to, we can say, fine tune the efficiency of the CAR T cells, at the same time try to take care of the uh, at the same time try to minimize the side effects. If you need to understand more about this advancement in the CAR T cell therapy, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.